Good morning, everyone. I'm delighted to welcome you all today. Thank you for joining this session. Today, we'll be exploring calculations of residual solvents. I'm Dr. S.K. Singh, and I'll be guiding you through this presentation. Over the next few slides, we'll cover the methods used to calculate residual solvents, their significance, and some practical examples. Let's get started and make this session both informative and engaging. Residual solvents are organic volatile chemicals that can be found in pharmaceutical products even after manufacturing. They either remain in the drug substance, excipients, or the final product itself. These solvents are used or produced during the synthesis or formulation processes, which means they can sometimes be left behind if not properly removed. In this slide, we're focusing on how we detect and analyze these residual solvents using gas chromatography. This technique is essential because it allows us to identify and quantify these chemicals accurately, ensuring the safety and quality of the pharmaceutical products we develop. Highlighting this method connects to the overall theme of quality control and regulatory compliance in pharmaceutical manufacturing. In this slide, we'll discuss the determination of residual solvents, specifically methanol, acetonitrile, and dichloromethane. These are common solvents that might remain in pharmaceutical products after manufacturing. To analyze them, we use a gas chromatograph equipped with a flame ionization detector and a headspace sampler. The reagents involved include methanol, acetonitrile, dichloromethane, and N-dimethylformamide, which help us prepare and run the tests accurately. Now, it's important to understand why we're measuring these residual solvents. Residual solvents are organic volatile impurities that can stay behind in active pharmaceutical ingredients, excipients, or the final medicinal product. Their presence depends on the manufacturing process, but regardless, we need to control their levels for safety reasons. Based on safety data, these solvents are classified into three categories: Class I, Class II, and Class III. Each class reflects the potential risk to human health, with stricter limits for those more toxic. When we analyze residual solvents using gas chromatography, we're able to detect and quantify these impurities with high precision. This helps ensure the product complies with safety standards and maintains quality. In summary, residual solvent determination is a critical step in quality control, ensuring that any remaining volatile impurities are within acceptable limits to protect consumer health. Let's walk through the preparation of the vial for the standard. First, we pipette 5 ml of the standard solution into a 20 ml headspace vial. It's important to close the vial immediately afterwards using a suitable septum and a screw or crimp cap to prevent any loss of volatile components. This standard solution's concentration is carefully prepared to meet the limits for Class II solvents as specified in IP 2018. Specifically, it contains 3,000 ppm of methanol, 410 ppm of acetonitrile, and 600 ppm of dichloromethane, all relative to the amount of test sample we're analyzing for residual solvents. Now, moving on to preparing the blank. We pipette 5 ml of N-dimethylformamide into another 20 ml headspace vial and close it immediately using the same type of septum and cap. This blank helps us account for any background contamination from the solvent itself during analysis. Finally, for the standard solution, we accurately weigh about 0.24 grams of methanol, 0.033 grams of acetonitrile, and 0.048 grams of dichloromethane. These are placed into a 100 ml volumetric flask containing roughly 25 ml of N-dimethylformamide. We then make up the volume to the mark with the same solvent. From this, we pipette 5 ml into another 100 ml flask and dilute to volume with N-dimethylformamide. This process ensures we have a precise, diluted standard solution that aligns with our analysis requirements. Overall, these preparation steps are crucial for accurate residual solvent analysis using gas chromatography, ensuring our results are both reliable and compliant with regulatory standards. Let's walk through the preparation of the vial for our test sample. First, we need to transfer about 0.2 grams of the test sample ensuring it's accurately weighed. This is then placed into a 20 milliliter headspace vial. Next, we add 5 milliliters of N-dimethyl formamide to the vial. Once that's done, it's important to close the vial immediately with a PTFE septa and a screw cap to prevent any evaporation or contamination. Moving on to the system suitability solution, we prepare this using a standard. This standard solution helps us verify that our system is functioning correctly before we analyze actual samples, ensuring the accuracy and reliability of our results. Now, let's discuss evaluating the blank. We place a vial containing just the solvent or blank matrix and record its chromatogram. The key point here is that we should not see any interfering peaks at the retention times of our analytes. If there are any peaks observed, we need to account for them by subtracting their areas from the peaks in our standard and sample chromatograms. This step is crucial to ensure that any signals we see in our sample are truly from the analytes, not background noise or contamination. All these steps, sample prep, system suitability, and blank evaluation, are interconnected. They help us ensure that our gas chromatography analysis of residual solvents is both accurate and reliable, ultimately supporting the overall quality control process. Now, let's walk through the procedure for analyzing residual solvents using gas chromatography. First, we start by placing 2 vials of a blank sample into the instrument and recording their chromatograms. This step helps us establish a baseline and identify any potential contamination or background signals. Next, we move on to placing 6 vials of our standard solution, which contains known amounts of solvents. Recording these chromatograms allows us to determine the retention times and establish a reference for identifying residual solvents in our test samples. After that, we add 2 vials of our actual test sample, which is what we're analyzing for residual solvents, and record their chromatograms. This is crucial because we can compare these results to our standards to see if any solvents are present. Lastly, we include a bracketing standard, just 1 vial, and record its chromatogram to help ensure the accuracy and consistency of our measurements throughout the analysis. Remember, the elution order, or the sequence in which compounds come off the column, is confirmed from the standard solution chromatogram by noting their retention times. This step is key because it helps us correctly identify the solvent peaks in our test samples. Finally, we pay close attention to the area counts of the solvent peaks in the test chromatograms. These counts are essential because they allow us to quantify the residual solvents present, which is a critical part of ensuring product safety and quality. All these steps together help us accurately determine the residual solvent levels in our samples, maintaining the integrity of our analysis and supporting overall quality control. Now, let's talk about the note regarding bracketing standards. It's important that the bracketing standard is injected either at the end of each new sample, at the end of a sequence, or after every 10 injections of test preparations, whichever happens first. This helps ensure consistency and accuracy throughout the analysis. Next, we need to calculate the content of each solvent using a specific formula, which involves measuring the areas from our gas chromatograph. The key point here is that the relative standard deviation, or RSD, of these areas, obtained from 6 injections of the standard preparation, should not exceed 15%. This is our system's way of confirming that the results are precise and reproducible. It's also worth noting that we can set the RSD at a value lower than 15% if we want even greater precision. Moving on to the overall system suitability criteria for residual solvents by gas chromatography, the main takeaway is that the RSD of areas from 6 injections should be within that 15% threshold. This ensures our system is reliable and our results are consistent. So, to sum up, maintaining this RSD limit and following the injection and bracketing protocols are essential steps to ensure that our residual solvent analysis is accurate and reproducible within the established standards. Now, let's talk about how we determine the solvent content in our samples. The key point here is that the solvent content is calculated as an average of the results obtained from 2 separate headspace vials. This approach helps ensure accuracy and consistency in our measurements. For each sample preparation, we analyze both vials, calculate the solvent content from each, and then take the average to get a representative value. This method minimizes variability and provides a reliable estimate of the residual solvents present. Remember, the goal of this process is to accurately quantify residual solvents using gas chromatography. It's a critical step in ensuring our product safety and quality. By thoroughly calculating and averaging these results, we can confidently report the solvent content, which ties back to our overall theme of quality control and compliance. Let's take a closer look at residual solvents and how we analyze them using gas chromatography. Residual solvents are the traces of solvents that might remain in a product after manufacturing, and their presence needs to be carefully monitored for safety and quality reasons. Gas chromatography is the preferred method for this because it allows us to separate, identify, and quantify these solvents effectively. The process involves vaporizing the sample and passing it through a column, where different solvents will separate based on their chemical properties. This technique is highly sensitive, which means we can detect even tiny amounts of residual solvents, ensuring our products meet safety standards. Overall, using gas chromatography for this purpose underscores the importance of precise analytical methods in maintaining product integrity and consumer safety. For further support, please don't hesitate to reach out. If you have any questions or need additional information, I'm here to help. Remember, we're committed to ensuring you have all the resources you need to succeed. Thank you so much for your time and attention today.